Yeah. Thanks for everything, John.
Anything you want me to say about you?
Nah, let's keep it simple. Hey, Jason.
Do you have a drink or anything?
I got my coffee, so I'm good. Thanks, John. Oh, yes. Okay. There we go. Okay.
All right, good afternoon, everybody. So this afternoon we are here for the Dissertation Defense of Eric Ma. And our procedure for the time period is going to be the following. We'll have Eric present his dissertation work, during and after which we'll have time for questions from the general audience. Then I will ask you all to leave, and Eric will have a closed session with his committee before we complete. So, by way of introduction, Eric was an undergraduate at the University of British Columbia, where he earned a Bachelor of Science degree with distinction in integrated sciences. And while at UBC, I understand he was advised by, amongst others, Brett Finley there. And I suspect that maybe that was the beginning or part of the seeds that were planted for his interest in infectious disease that he ultimately ended up pursuing here. Ultimately, Eric joined the graduate program in biological engineering here at MIT. And after spending some time exploring research areas, I was really happy that Eric decided to join my group to complete his graduate work. Eric is somebody that, from my interactions at the time and from my knowledge of his history, struck me as an inherently inquisitive person and somebody who was not afraid, and in fact was more than willing to engage in problems and discussions across a really wide range of topic areas, and that included some of the things that we were doing, fortunately. I think that sort of spirit has borne out during Eric's time here, and during his graduate work, and I want to say that, in my opinion, and certainly for me, Eric embodies one of the real joys of being a faculty member, and that's the opportunity to engage in reciprocal learning interactions with scientists, and particularly young scientists like Eric that have that real spirit of learning and inquisitiveness about the world and the energy to go about doing it. I've learned a lot from Eric over the last few years, and I know that he has enriched the academic and personal lives of those around him. I think that over the last few years, Eric has learned a lot during his graduate work, and fortunately he's here to share some of those achievements on the way to completing his dissertation. So Eric's title for today is Identification of Reassortment Influenza Viruses at Scale, Algorithm, and Applications. Eric.
Okay, thanks, John. Thank you, everybody, for coming. It's my pleasure. I just wanted to start by saying I've learned a lot, much more than I think I've shared with other people. And so came into this work not knowing much about evolution, ecology, and stuff, and I've learned so much from the lab about the fundamentals of evolutionary ecology. I came into the lab not knowing computation very much as well, and through interactions with the committee and your groups as well, I've learned so much about computation. So a big, big thank you to everybody for all the support here. So I'm going to talk a little bit about flu, right? And we're interested in flu because of the damage that it does to human populations. It causes billions of dollars of damage. It causes loss of life. But flu is much more than just a pathogen. From a scientific perspective, it's very interesting as well. It's a wildly, insanely successful virus. It's been able to infect all sorts of hosts beyond humans, chickens, and pigs that are the hosts that we care about the most, right? Its reservoir host is wild birds, ducks that fly all around the world. It's infected all sorts of wild, big animals, right? Seals, camels, penguins, whales. You know, you think of a big animal, and you probably should be able to find flu over there. In addition to that, new flu viruses are being found in new types of viral hosts. Bats, for example, which are thought to be a reservoir for other types of dangerous viruses. Now we've found new viral influenza subtypes in bats. Part of the reason why flu is thought to be so successful is because it's got this capability to hypermutate. It's a really fast evolving virus. And that stems from it being an RNA virus. But also, part of its ability to be so successful stems from its ability to shuffle its genome, right? And so reassortment is what we call genome shuffling in the flu world. And it stems from the fact that flu has a segmented RNA genome. If you take, say, a blue virus and a red virus, and they let them co-infect the same viral host, the progeny that can come out can be a mixture of red and blue, can harbor a mixture of red and blue segments. Actually, I've got 3 printed models for the audience, and if the science gets too distracting, if the science gets too dense, you can start exchanging genes with one another, all right? So it's thought that reassortment is also capable, it also contributes to its ability to infect this wide range of hosts. So if you go back, and let's bring the focus just a little bit back to humans again. If you think about the pandemic viruses that have infected humans over the past, say, 100 years, for which we have sequence data, and you trace back its evolutionary history over time, you'll find that you'll see a virus that spilled over from animals and humans. Part of its evolutionary history involves a reassortment event that gave rise to this new virus that spilled over and started causing trouble for humans. So this gives rise to this common dogma. Oh, okay, reassortment should be implicated in pandemics. Maybe it's also implicated in host switches, because that's the most common thing that happens when we have new pandemics. But we've never been able to test this idea in a systematic fashion. And here's why. The best algorithms for finding these shuffled viruses are predicated on the construction of phylogenetic trees. And these don't scale very well. And so the contribution that I've been fortunate enough to make to the scientific literature here is the development of a new algorithm for finding these shuffled viruses at a large scale. Let me very quickly run you through the background knowledge for the traditional way that it's done. We typically take our viral genes, and then we construct phylogenies of them. We're essentially trying to understand what the life history of the virus is. And there's some terms that I want to highlight on phylogenetic trees so that you can, to help you understand the later portions of this talk. The first thing that I'd like to highlight is this notion of internal nodes. When we construct a phylogenetic tree, we're doing hierarchical clustering of the sequences according to some model of how the sequence evolves. And we have to reconstruct these or estimate the internal nodes of these viral sequences, which then give rise to the structure of the tree. The second thing that I want to highlight is this notion of a clade. So this notion of a clade is what I'm highlighting over here. A clade is, simply put, just a cluster of viruses that are distinct from other virus clusters on that tree. And it may be something defined statistically, or it may be defined in a subjective fashion. But that's the concept there. And the third final concept that I want to highlight is this notion of a patristic distance, which is the sum of branch lengths from one virus to another virus, right? And so with these three terms, I'm going to show you now an actual phylogenetic tree taken from another study that was published a while ago now, 1999. But it's a study that investigates this phenomena of reassortment, right? And so on this phylogenetic tree, you can see we're looking at the internal nodes. We're estimating the structure of the internal nodes given the sequence data. We have the clades that are identified. We can identify clades on the tree. And the final part here is we can compute patristic distances between these viruses and use that as a metric for evaluating how phylogenetically distant two sequences are on the tree. Okay, so here's how reassortments are identified using this exact example over here. I've highlighted with a red box the viruses that are of interest. These are the viruses that say were newly sampled by the authors of the study. They're investigating, they want to investigate the phylogenetic history of this virus or this cluster of viruses. If I overlay, now I've also highlighted here the host species from which these viruses were identified. So we have the avian, the human, and the swine. And reassortment events are detected as such. You'll notice that over here, this cluster of viruses for the particular gene PB2 clusters with a bunch of avian viruses, whereas this cluster over here, this set of viruses of interest, clusters with swine viruses. And so what we'll say is that this particular group of viruses that we've newly isolated, they're the product of reassortment between an avian virus and a swine virus. And that gave rise to this new virus over here. All right? Taking a simplified view of this, it looks something like this. For example, I have avian viruses and human viruses, or avian isolated viruses and human isolated viruses, and I have my virus of interest, and I want to identify how this virus came about. If I come up with these three phylogenies for each of these three genes, then I'll say that this virus of interest was the product of reassortment between a human virus and an avian virus. All right? So there are mathematical definitions for how these viruses that generalize this rule of phylogenetic incongruence. And I'm not going to go through them in detail here, but I just want to put it out there that the mathematical definitions are out there. They're written in my thesis as well, and you can take a look at it. But essentially, the main problem that I wanted to highlight again was that these algorithms are conditioned on having a phylogeny reconstructed. And the phylogeny that's reconstructed takes exponential time with the number of sequences that are put into for phylogenetic reconstruction. And so that basically means that if I want to use phylogenetic tree-based methods for identifying these shuffled viruses, I'm going to spend approximately 90% of my time on tree construction and only 10% of the time on identifying these incongruencies. And that time is exponentially growing with the number of sequences that we input. So can we find a better way, a faster way of approximating the phylogenetic reconstruction? That's the quest that I set out to go on. So when I started work on this, I was looking around and I was trying to figure out, can we use a network-based representation? Rather than look at trying to estimate internal nodes and trying to estimate what these hidden states look like, can we just take an approximation and say, where did this virus come from? And the inspiration came from this paper that was published in 2011. And I fondly remember I read this paper about 6 or 7 months into starting this work. And I looked at it and I was going, shoot, I'm scooped. Someone already did this. And I immediately thought back to Uri Alon's scoop little ukulele thing. But then later I looked at that paper in a little bit more detail. I realized, no, I'm not scooped. The best part of this was Dr. Jambart did all the hard work of convincing people that we can use network representations, and so I don't have to. I can do the lazy man's thing and work based off his work. So his algorithm effectively became the inspiration, the grounding inspiration for what I did. So Thibault's algorithm basically works like this. Imagine if you have a bunch of viruses listed and you order them by time and you sort of layer on metadata over there. What we're trying to do is essentially ask, where did a virus come from? So I look back in time and I say all 8 of my segments came from that virus. So I'll draw a line from, because that other virus looks most similar to me. So I'll draw a line from that virus to me. Then I'll go on. For this other virus, look backwards in time and ask which other virus looks most like mine, right? And that's the essential core of the algorithm. Now, the place where we made a modification was this. If I find a virus for which every other virus back in time just doesn't plausibly look like a clonal transmission or clonal descent of all 8 segments, maybe what I should do is split up the search and look for source pairs instead. So part of the genome came from here, and part of the genome came from there, and that results in the identification of these reassortment edges, right, in the network. And so clearly what I've given you is just the conceptual understanding. There's a lot of little tweaks that ended up in the final work that I'm not talking about here, and I'm happy to discuss these later on as well. One thing that does come up is a lot of people will ask, are you going to force the identification of a source for every single virus? And the answer is no. There are still singletons that are possible, and these are viruses for which we don't have a plausible explanation. And I just wanted to put that out there in that we're not trying to impose and say that every single virus has to find a single source or a source pair, all right? Okay, so now I remember the 25-minute lecture during my first committee meeting about the importance of simulation studies, and so we conducted these simulation studies. That was the time. That was part of the lessons that I've learned about computation. If you develop a new algorithm, make sure you validate it with simulations. So I took that lesson, implemented a very simple small world simulation, and found that in essence, the essence of the idea here is small world simulations can do a better reconstruction compared to a null reconstruction. And the null reconstruction is a random search for sources back in time. And that, for me, was good enough for simulation purposes. As a side story, I was asked to review a paper for PLOS computational biology, and the authors forgot to include simulation studies. So JP, Mark, that lesson has been passed on. Okay. But for me, the simulation studies weren't enough. Accuracy and identification wasn't enough. One of the claims we're making here is we're actually able to approximate phylogenetic reconstruction well. Now, it was sort of established in Thibault's paper, in the original 2011 heredity paper. But I wanted to go a bit more quantitative than what they did. And so what we've done here is taken a look at the patristic distances that are captured by the phylogenetic, by the network reconstruction. To get to this part, this set of figures, I'm going to guide you through a bit of the logic behind that on the blackboard. Okay, suppose I had a phylogeny that looked something like that on the blackboard there. When I say we're looking backwards in time and asking who's most similar, essentially the hack here is I'm drawing a line, oops, wrong way. I'm drawing a line from one virus to the next. The patristic distance that I'm capturing here should be as small as possible on the phylogenetic tree. If instead I drew an edge from here to there, then the patristic distance that I'm capturing is going to be longer, right? And so essentially what we want to do is to be able to capture short patristic distances on the phylogenetic tree using the network reconstruction method. And so in blue, in the blue columns, what you're seeing is for every gene segment for a subset of the data for which we constructed these phylogenetic trees, that's the full pairwise empirical distribution of patristic distances that are present for this subset of data. And in red, this is the subset of edges pairwise patristic distances that we're capturing using the phylogenetic approximation, the network approximation. All right? And so this, to me, was much stronger evidence that, yes, the network reconstruction is capturing good phylogenetic approximations, is doing a good approximation to phylogenetics. And so this gave us the confidence to move forward and do this on a large scale. The final thing about the algorithm that I want to mention is that once we've got the sequence relationships reconstructed, we can then move on to doing metadata analysis. Now, I've said this joke a number of times now. The NSA loves metadata, and I love metadata. I truly do love metadata, but I'm not trying to be the NSA here. So if we layer over metadata on the network, right, we can start to do analysis of the metadata conditioned on the underlying structure of the network. And this is where the powerful stuff, or the very fun stuff, I should say, this is where the much more fun stuff scientifically comes into play. And so that's the core of the algorithm. Like I was mentioning, there's extra details, but I'm happy to discuss this later. I'm going to talk now about 2 applications of the algorithm to studying scientific questions. All right? So the first application is this study of reticulate evolution, or let's just call it reassortment for now. Study of reassortment in flu and its relationship to other aspects of flu and viral host ecology. So if we go back to that dogma, reassortment is important for host switches. If we have a network reconstruction with all of the metadata layered over it, we actually now have an opportunity to quantitatively test this principle. All right? And so here's how we go about doing it. Just to give you a flavor for what the data set looks like, this is roughly 35 years of sequence data collection. It's a global effort. It's all deposited in the influenza research database. The analysis was conducted on 18,632 fully sequenced influenza viruses, amongst which we identified 2,352 reassortant viruses. And so in order to understand the following figures that I'm going to show you, I do need to first show you this empirical statistical test that we developed in order to, that formed the grounding of our conclusions. So this statistical test is essentially a test of whether pairs of node labels are associated with a particular type of edge, right? And so the pairs of node labels could be same host species or different host species, and the type of edge would be reassortment or clonal descent. So what we have in our data, say we had this data that's up here. We have all the viruses. They're colored by their host species. We have the edges, and they're colored by their type, clonal descent or reassortment. We can ask first, between pairs of nodes that have an edge between them that are of the same species, what fraction of edges are clonal descent and what fraction are reassortment? And we ask then for pairs of edges, pairs of nodes that have an edge between them that are between different species, what pair are clonal, what pair are reassortant. But we don't compare these 2 numbers directly. We compare them to a null distribution in which what we do is we permutate the host species labels across the entire data set, essentially breaking the relationship between these pairs of node label pairs and the particular edge types. And that gives rise to these 3 plots, where in light blue we see what we observe in the null model, and in dark blue we see what we observe in the data itself. All right? So let's go through the first one. The first figure is essentially the answer to the question: is reassortment overrepresented when crossing between different host species? And the answer is yes. Take a look at that bar on the left over here. And it's underrepresented between pairs of hosts that are same. The next thing is this notion of ecological type. Now, I don't want to go too much into detail here, be pedantic about it, but basically we can group viral hosts into human, wild mammal, wild bird, domestic mammal, domestic bird. Now, if we slice the data, we're going to get reduced statistical power due to reduced numbers, and so there's some thresholding criteria. Again, happy to discuss this later, but I just wanted to highlight that this principle more or less overall holds when considering not just host species pairs, but now host ecological type. right? So for example, what I'm highlighting over here is that where the ecotype between, say, domestic bird, domestic mammal, for example, where the ecotype is different, reassortment is overrepresented. And what I'm showing next is where the ecotype is the same, reassortment is not overrepresented. And what I'm showing over here now is the exceptions to the rule. All right? And so the exceptions are as follows. This first one over here is the ecological interface between humans and domestic mammals. And for the most part, we're talking about humans and pigs. Right? And it just turns out humans and pigs share a particular viral cell surface receptor, meaning clonal transmission between viruses and pigs is actually greatly enabled. And so reassortment, for that reason, probably is underrepresented because of that. The other example is wild bird to wild bird, which, as you might imagine, actually encompasses a lot of eco-subtypes, lots of different wild bird species. So this is partly an artifact of how we group the viral hosts together, and also because of the ecological sub-niches that each of these birds play. All right, so that's that for the ecotype. This is where the fun part comes in. If we think in an abstract and a global level about how each of us viral hosts can form an ecological niche, think about it. Humans can, at an abstract global level, be considered one ecological niche for the virus. Chickens in farms can, at an abstract level, form one ecological niche for the virus. And so where we have the data available, we can actually start quantifying the distance between these types of ecological niches. And one way of quantifying this is to use the evolutionary distance as represented by the cytochrome oxidase 1 gene. Now, this is not the only gene that you can use. There are much more refined ways. And again, I'm happy to talk about that later. But here, the cool idea that emerged from this analysis is that as the ecological quantitative distance between viral hosts increases, the proportion of reassortment also increases. And that seems to speak to the difficulty of crossing between viral hosts, crossing between barriers, ecological barriers. And it may be that where the ecological barriers are high, this ability to shuffle the genome becomes extremely important for being able to cross this. Now, we might say, yeah, that's sort of kind of intuitive. And yes, it is intuitive, though it's never been tested systematically. And this now is, I think, one of the biggest contributions that we're able to make from this model, from taking flu as this model sort of microbe and taking the reassortment as this model for gene shuffling and other reticulate evolutionary processes. So there are some limitations to the study. I just wanted to give you all a flavor of it. I won't go too much into it. The first limitation is we're never really identifying whether reassortment happens before host switching or after switching. This question has come up so many times. And truth be told, I don't have a very solid answer as to how we can modify the algorithm to identify before and after. I think we need better sampling, denser sampling, and co-sampling at ecological interfaces to identify whether viruses jumped first or reassorted first. All right? The second thing that always comes up with any global analysis of publicly available data sets is sampling biases. And again, the gold standard thing to do would be let's increase sampling of wild birds. Well, let's also increase the NIH budget so we can do that, right? So we need other ways to get around it. And one of the ways that we did do this is we did do this pseudo-sensitivity analysis in which, if you remember the permutation test we just shuffled the host species labels. The pseudo sort of sensitive analysis test is where we equalize and then shuffle, right? And we ask, do the conclusions change? And the answer is the conclusions don't change. If you want some technical detail, means don't change, variance changes, and even when the variance changes to be really high, reassortment is either still really overrepresented or it just never was overrepresented to be in. So the conclusions basically don't change. I also wanted to expand everybody's horizons a little bit. Think about, we've talked about flu all this while, but does this apply to other viruses? Anecdotally, maybe yes. Think about HIV. HIV, we know it has monkey origins, monkey virus origins. This is something that's sort of generalizable across a principle that now we can possibly have a way of systematically testing across different domains of life. All right, so with that, that sort of concludes this story. This study was published last year. It, for me, is one study that's very close to my heart because it's my first contribution to scientific literature that I was able to take from completion all the way to execution, all the way to, from conception to execution to completion. Sorry, got that wrong. After the fact completion. From conception to execution to completion. And one of the nice things that emerged from this is that the software that I wrote, which currently exists as this one giant hack in GitHub, it's actually under consideration for being used at the influenza research database, and that's a very nice thing from a software engineer standpoint, that the software that I built, somebody wants to use it. Just one person's good enough. Okay, so that was the study of flu at a global level. And here we were talking about the interplay between at a global scale. So now I want to talk about the application of the algorithm in a study that was much more focused, that also still speaks to this idea of host-virus interactions. All right? So this second application is viral transmission and host migration dynamics. The sub-question is, do viral transmission strategies follow wild bird host seasonality? And just to give you a flavor for this, first off, when we talk about viral transmission strategies, again, on an abstract level, we can think about it as being clonal. Basically, I go to you, you go to the next person, and spread like wildfire. Or we can think about it as, let me find another gene to incorporate and let me find one gene to throw out and find another gene to incorporate. So let me reassort with someone else. Right, so this we can think about it as being a transmission strategy with quotation marks. So if you think about then also the behavior of birds, the wild bird reservoir. Wild birds, they have two stages, breeding and feeding. I call it breeding and feeding. Maybe Nicola might call it something else. But I sort of think about it as breeding and feeding. Breeding is in the summer, feeding is in the winter. Breeding is in the north, feeding is in the south. That's sort of the way I think about it at a high abstract level. There are a lot of details I'm sweeping under the rug here. So then we can ask, does reassortment happen more during breeding, or does reassortment happen more during feeding? Or is it the other way around, right? We can ask these questions given an algorithm that is capable of finding these reassortments, and given other metadata that we layer on top of that. So this was the part on, this was the part on showing where the birds fly. Just so that you have a bit of sense of geographic scale, this is the sampling site. Birds generally move from here to here, from June, July, August, September, November, then they, it's this way. June, July, summer they're there, winter they're here. And yes, and winter they also go that way, all right? So that's the big idea there. The data set. Now I really want to say a bit about the data set because this data set is very, very, very tough to come by. It's very valuable. 3 years of sampling, dense sampling to get from 14,000 wild birds, all right? In Minto Flats, Alaska, yielding 545 plus 1,200 more viruses. So 545 viruses came out from 14,000 sampled birds. And we added on 1,200 plus more publicly available data, all right? And sample collection is non-trivial. I fully appreciate that being in the Runstadler lab. Like, I'm just the computer guy, right? There's a whole bunch of people doing logistics and spilling sweat and blood out in the field, risking mosquito bites, playing with fire rocket launchers to launch nets onto birds. There's a lot of stuff that goes into collecting this sequence data, all right? So big salute. Thanks a lot, Nicola, for the photos as well. It really brings home the point. This is a tough job for the field biologists there. But some people like it. Salute to them. So, okay. So let's go back to the scientific questions. You have the breeding and the feeding stages. You have reassortment and you have clonal transmission. Do they show up at different times, all right? So we can interrogate this by looking at the spatial distribution of edges in the network. The spatial distance between edges in the network and the temporal distance between edges in the network, right? So here's an example of studying the spatial distribution. On every single viral node, we have the GPS coordinates of sampling or the state of sampling. That allows us to compute the distance between the start or the source virus and the sink virus, okay? And so if we look at just the latitude, if viruses start and end at the same latitude, then they're going to fall along the diagonal if we plot source and sink against each other. And likewise for the longitude on the right-hand side in purple. So if we break down the edges in the network by full complement or clonal descent versus reassortment, we'll see that clonal edges tend to fall along the diagonal, implying that they're generally quite local. And reassortment edges tend to follow long geographic distances, implying that they are more spatially involved in spatial dispersion. And this probably follows the annual cycle. This is a hint that it follows the migratory patterns of wild birds. If then we take a look at the temporal part of this, again, we can take a look at the the source date and the sink date, plot them on a two-dimensional plane, and if they fall along the diagonal, then they're temporally local, and if they don't fall along the diagonal, then they're temporally spaced apart, right? And so that's what we see so we see that clonal descent edges, which is the top panel of the left-hand side over here, clonal descent edges tend to follow diagonal over there, whereas reassortment edges have a greater presence. They also still tend to follow the local, they also have this strong local component, but they also have a higher representation in the off diagonal, right? And if you look at then space against time simultaneously, you plot in orange the clonal descent edges, you'll notice most of the clonal descent edges are at the Minto Flats latitude, and the reassortment edges, as time progresses, follows with the bird migration as they go down to the lower latitudes. So it looks like reassortment is playing a role in viral persistence, or it's following, at least it's following the bird's patterns as the bird's behaviors change. Just to get a different view onto the temporal nature, right? Over here, I've plotted every single viral isolate on the x-axis, and we draw arcs between them to show the temporal relationship between the viruses on the network. And again, it's clear that it's local temporally when we talk about full clonal descent edges, and it's much more dispersed temporally when we talk about reassortment events. So this work was much richer than the two-figure panels that I'm showing over here. Big credit to Nicola for leading the work and getting it published. It was a very hard struggle. There was like 4 reviewers, and reviewer 4 was tough to convince, but we convinced them at the end. We fought through. So that was a nice thing. It was a nice thing to happen, and it really again helps. From my perspective, it's sort of intuitive that the virus, being a parasite of the bird, should follow its behavior, follow the bird's behavior as the bird's behavior changes. But again, this has never really been tested. It remained intuition. And I think the contribution of the reassortment finding algorithm was basically we're able to identify these reassortment events. Even though it was at a smaller scale, it was at a denser scale, and we were able to map with finer resolution some of these questions. Okay, so this is the final part. There are some open questions that remain. I don't think anyone in grad school can ever claim to have solved the field. And so these are the open questions that I'd like to highlight, maybe as an inspiration for someone else who might want to take on this work. The first one was this idea from the First, global study. This idea of quantitative ecology, quantitatively identifying differences between ecological niches. They're traditionally identified in a qualitative sense, categorically. This thing feeds on that guy. This guy predates on that other thing. But what if we could start quantifying differences? What's the quantitative difference between the prey? What's the quantitative difference between this niche and that niche? And I think those are, that might open up a new world of statistics, statistics in ecology. The second thing that I think is a very pertinent one for influenza particularly is the forecasting of the emergence of new viral subtypes. As I mentioned before, reassortment is the one that, reassortment is the process that's implicated in all of these pandemic outbreaks in humans. And typically, they're reassortment of the external genes to which we generate immunity. And so if there's a new subtype that comes up, well, boom, we don't have immunity. So good luck to us. So what if we can find a way to forecast the emergence of these new viral subtypes? That might be a new cool question to tackle. The third thing comes from this notion of mixed infection, right? And this might help us towards answering this question of whether reassortment happens before or after the host switch, right? When you have a mixed infection and you sequence that sample, the unfortunate part about the data engineering that goes into the IRD is that you have to report one sequence, one subtype, one host. What if we found a way of firstly reporting multiple sequences at the same time from a single sample, and number two, this is the more crucial part, phasing the samples, right? Just as we do genome phasing to parental on human genetics, what if we found a way of phasing the genome segments the mixed genome segments to the rest of the population context that's out there, right? That might give us a way into identifying these reassortment events, and that would give us a way into identifying the co-infection events that happen before the reassortment events that happen before then or after the host switch events, right? And so this would give us greater resolution there. Okay, final part is I've learned so much about software development and software engineering during my time here. Again, like interacting, Mark, JP, with your groups has been really helpful, and going out to PyCons and learning from the best software engineers out there has been really helpful. And good software gets used, and I'm really thankful that I've been able to develop something that at least someone's willing to try to think about using. Even if they don't use it, that's okay. I'm happy that they at least considered it, right? Because not everybody gets the privilege of having that consideration. And so, and good software is actually going to be increasingly important for reproducible science, and being able to publicly archive our work and documenting the software properly is going to be really important. So if any of us here ever get into software work, please do it in a reproducible fashion, release your work publicly, and develop good software that people will use. And with that, I want to thank you all for your time. It takes a department to raise a grad student, just as it takes a village to raise a child. And so I couldn't have done it without my colleagues, collaborators, mentors, as well as other students who've enriched my life and during grad school as well. Thank you very much. Happy to start a discussion now if you all have any questions about the work. Mia.
I have a question.
Yes.
Do you have information on the temperature, the environmental temperature, or some environmental variables when these samples are collected? Because especially with climate change, that's going to
Oh,
change
yeah.
patterns. And is there any effect on influenza reassortment based on temperature? Because it seems like that algorithm helps predict something.
Possibly. Actually, yes. It looks like that could be a piece of metadata that could be overlaid, because we do have not just the host species, but also the geographic geographic location, at least at the state level, right? We stab it at the state level or county, not really. State level we do, country level for the rest of the world. And so it's possible, I think, to go back into historical data and then start mapping out temperature change as a function of time versus reassortment events that we identify as a function of time. And so yes, that could be a possibility. Could definitely be a possibility, at least at the global scale. Global scale. Claire.
Yeah, thanks. Great talk here. So as someone who doesn't really think about birds or viruses ever, my question might be basic, but I'm really interested to hear, right,
Yeah.
Why would we expect it to be different, and what does it mean about,
If it's okay, I'll try to answer this from a public health perspective, because I know that's something you're interested in. So biosecurity at farms is one big thing. And then, and wild birds can release viral genes into farms. All right? So the biosecurity of a farm is one thing that we can tackle on its own. But another thing that we can tackle is then, if the bird's annual movement follows this cycle, maybe we can start looking at, actually it ties into a little bit into this forecasting of new viral subtypes. If during the breeding stage it's mostly clonal and there's just wildfire spread of virus, but now when you come to the lower 48, there's now this gene pool mixing that's ongoing. Maybe what we can do is we can start looking at what gene pools are going around, and in addition to having better biosecurity on its own, also think about, can we vaccinate the chickens against this new viral subtype ahead of time, right? Or have a repertoire of vaccines that are animal vaccines that are ready to go prior to the emergence and spillover. And so if one event starts to happen, we can just quickly ring vaccinate around that farm and stop the spread of this new viral subtype that comes in. So that might be an application there of understanding the seasonality of this viral movement. Bill.
Where all this variation comes from. Just summarize those of us who don't know.
Yes, absolutely.
For instance, if you have an RNA, or an RNA polymerase is a track error, or is an RNA DNA replicated intermediates?
Right.
DNA, and where does this come from? And how fast is it?
Right, right. Absolutely. So influenza, I'll give you a sense of scale here. If you look at the human, it's a 566 amino acid protein, and every year you'll see about 3 to 7 on that order number of amino acid changes if you look back at the historical data. And then
In general, you get some mutations take place again and again and again.
some mutations do take place again and again, especially if,
When they're not under any selective.
oh, pardon me,
When they're not under any selective.
do they?
That's hotspots.
Ooh.
You see them in observation here. I didn't hear the idea that certain mutations are going to be expected higher than chance. It sounded like we talked about they all occurred in some undefined but similar development.
Okay. Here is basically one, are there hotspots of mutations? And my answer is I'm not very clear. It's possible. I know how to do the analysis for that. I just haven't done it. And so my answer is I wouldn't be sure. My naive prior is the hemoglobin pressure. Those are the ones where we would expect hotspots. The internal genes, not really, right? Because it's not really under selective pressure except to function, right? The only thing that they have to do is function. They don't have to beat an immune system on the outside. As for how this variation is generated, it's because the flu carries its own. And the fifth genes constitute the RNA-dependent RNA polymerase. And for that reason, well, okay, I think for me, it's merely a coincidence that RNA-dependent RNA polymerases are sloppy. It's not that they're sloppy because they're RDRPs. But yes, they are RDRPs. They're sloppy. And so they're going to incorporate a lot of mutations.